I'm here today to introduce Dr. Tristram Wyatt. He's a Senior Research Associate at the Department of Zoology at University of Oxford. He's also the founding fellow of Kellogg College, Oxford, and the founding chair of the LGBT Oxford Staff Network. He also refounded and reinvigorated the LGBT network at Imperial University. He's published quite a few books. The first one, Pheromones in Animal Behavior, Communication by Smell and Taste. And his second one, Pheromones in Animal Behavior, Chemical Signals and Signature, won the Royal Society of Biology Prize for Best Postgraduate Textbook in 2014. He's featured quite extensively in his TED Talk and additionally on the BBC. His most recent book is Animal Behavior, A Very Short Introduction. So if we can ask Tristram, how did you get involved in pheromones?
Well, I needed a job, and I'd done a rather obscure PhD, which was all about parental care in beetles, which was fascinating, but it wasn't a job in demand. And pheromones are incredibly important for lots of animals that eat crops, so especially moths. And we know a lot about moth pheromones, so there were some fantastic jobs going. And that converted me into reading about pheromones and then never giving them up.
I look forward to hearing a little bit more about what pheromones exactly are, so please,
Great.
tell us.
Well, thank you very much for coming. Happy Pride. Looking forward to getting out to the march after this. So I work on pheromones, which are these invisible chemical signals between animals of the same species. And we've known about them for a very long time. The ancient Greeks knew that female dogs signaled that they were ready to mate to male dogs by a smell. And you could take the smell from the female dog onto a towel, and the male dogs would be misled and would chase the towel rather than the female. So we knew it wasn't a special bark, it was a smell. But the problem is that animals produce vanishingly small quantities of these pheromones. So picograms, micrograms, it's incredibly hard to study. And it wasn't until the 1950s that the first pheromone was identified. And that was in the silk moth. And the team that worked on it had spent 20 years and made a very wise choice. Because with each female producing a tiny quantity, the researchers needed 500,000 moths to get enough material. And the thing with silk moths is that we grow a lot of them, so they had plenty. Now, before I go any further with pheromones, I ought to talk a little bit about what pheromones are not. And I don't know how many of you have been to a pheromone party. It was advertised in Time Out, and basically you wear a t-shirt overnight, and then you go to the venue, having taken off your t-shirt and put it into a plastic bag with a color-coded number. And then at the pheromone party, so-called, you open the bags, sniff the t-shirt, and then if you like it, you take a selfie. And you can choose same-sex t-shirts or opposite-sex t-shirts, or both, depending on what you like. And then the idea is that your selfie goes up on a website, a private website, and if you like the look of the person who's taken the selfie, it breaks the ice and you meet over a glass of wine or a beer. Now, this is all about smell, and it does break the ice very effectively. And I have to say that my husband actually does smell wonderful, but we didn't meet there. But the thing is, this isn't about pheromones, because pheromones are molecules that are the same in every female or every male. And what that party is doing is looking for differences. And the idea behind it is that you are looking for somebody who's genetically compatible to you, and somehow, for reasons we don't understand, your immune system changes what you smell of. But that's all about individuality and compatibility. That's not pheromones. And of course, the reason we can find pheromones is when those researchers were looking in the silk moth, females, they were looking for a molecule that was the same in every female. And that's how they knew that they'd found the pheromone, and they could ask the males to make sure, because the males would respond by fluttering their wings and getting very excited. So that's all about what pheromones are not and what pheromones are. And since that first discovery in 1959, they've been found all over the animal kingdom. So insects, fish, it works just as well underwater, lobsters, mice, deer, lizards, lots of fruit flies. So basically, any animal you can think of is likely to use pheromones as part of its communication. And it's not just about sex. It's also about all the different kinds of ways that social insects like bees and ants and wasps organize their lives. And it's pheromones that are behind the problem of ants at picnics. So the ants send out scouts. They don't know where they're going, but when they find your picnic, they rush straight back, laying a trail of pheromones, and now the other ants in the colony can come out and find your sandwich. So that's how it works. And the same in the way that the whole of the honeybee hive is organized. So that's why the workers don't reproduce, because the queen is signaling that she's there. It's how they organize the defense against bears and humans. So if one bee stings you, then other bees will be attracted by the pheromone left on the sting. And so that's why it's a good idea to pull out the sting if you've been stung once, to avoid other bees coming in to sting you. So apart from that practical advice, what I want to move on to now is the sad story of bad science as concerns human pheromones. And that's something that is of great interest to me, because I've been talking about the rest of the animals in the animal kingdom, and I think that probably humans do have pheromones. It's just that we haven't found any yet. Now, if you Google pheromone, you'll find more than 1 million hits, and most of them are trying to sell you some human pheromones. And if you go to the websites, you'll find there are lots of people wearing lab coats, and the names they give to the molecules are long, like androstenone, androstenol, estrotetraenol. So there are lots of ols, and it sounds really scientific. It got me curious. What was really going on here? Because there's also a very big scientific literature. There are more than 40 studies on these molecules that claim they found a big effect of attraction, on emotion, on all sorts of things. So I started doing some detective work. And what I found was astonishing. If you go back to the 1991 paper that establishes these two particular molecules, androstenedione and estrotetraenol, as being the human pheromones, the method section of that paper is just 4 words. The molecules were supplied by the Erox Corporation. Now that is not a method section, and you'll know from the rest of the exhibits here that if you're going to do science, you have to do it properly. So that really doesn't count. And what it emerged was that simply 2 very enterprising professors at the University of Utah, the home of cold fusion, that was different professors, these professors had patented these molecules as human pheromones, but they never provided any evidence. So what we have is a ground zero of a paper claiming that these molecules were pheromones. Nothing might have happened, but in 2000, a leading researcher published the first paper using these molecules and doing what looked like a really nice study. Because they were a big influence on the field, everybody else thought, okay, it must be true. And what we have is a whole series of papers citing that first 2000 paper and never going back to that very first one to find out what was really there. And it's part of a very interesting phenomenon of an echo chamber where people look for an effect and they only report it when they find it. And if they don't find something, they don't publish it. It's called in the field a desk drawer effect, because the results go in the desk drawer. Now, what really impresses me is the experimental psychologists have been taking this on. So the experimental psychologists, and this, when you're looking at human behavior, that's really what you're doing. It's called experimental psychology, asking questions about human behavior. They've started to realize that they actually need to start again and do things properly, doing everything double-blind, declaring what results they're going to report, how they're going to analyze the results in advance. And that's a whole area of improving the reproducibility of it. So what I'm hoping is that people will now start to look at human pheromones in the way that they're looking at the rest of experimental psychology and start doing some really good work. Because Charles Darwin, who was a fellow of this society, recognized humans are just another kind of animal. And what he also talked about was the way that males get very smelly because of female choice. He saw it, just like the peacock's tail, as being a perfect example of sexual selection. So the smellier males get chosen by the females and probably vice versa. Now I want to leave you with just one more thing, which is studying human sexual behavior is really difficult. People don't always say what they're thinking, they don't always do what they're saying. There's lots of stuff that makes it hard, quite apart from ethics committees and getting funding. But there's one area that looks as though it might turn out to give us the first human pheromone. And this is some lovely work coming from Dijon in France. The lab is run by someone called Benoit Schall, and he works very closely with mothers and babies. And it looks as though newborn babies are responding to a smell given off by lactating mothers producing milk, and the baby turns towards the nipple that the smell is coming from, opens its mouth and starts to suck. Now that gives you a test for working out is the pheromone there or not. And the team has just got a grant to work with chemists to try to find the molecules that might be involved. So if we were talking perhaps in 5 years' time, we might, by that point, have a human pheromone. I think it'll be a lot longer for sexual pheromones to be found, if we find them at all. Thank you very much.
Thank you, Tristram. It was a really interesting part, and I was reading some of that article, and one of the things it talked about is the strength of the pheromone coming from those nipples can impact how well a baby can attach. And so are there different levels of pheromones that you can get in different animals?
So there are two aspects to that. One is that it does look as though, and this is a correlation rather than actually being properly shown, but there's a correlation between the number of the bumps around the mother's nipple. So the pheromone seems to come, the secretion seems to come from these little bumps around the nipple. And the more of these little gland bumps around the nipple, the faster the baby seems to latch on. And one of the important things about that is you'd think that a baby has only one job, which is to latch onto a nipple and get milk. But a surprising proportion of babies are not that good, which is distressing both for the baby but also for the mother. And so what would be great if we could find, or rather if they could find the molecule, is that if you could synthesize it and then put it into a kind of lipstick or chapstick, you could put it round the nipple and get every baby to latch on better. And that's really important because the first milk meal is important for all the colostrum and also just getting hydrated again.
Do you think that that's replicated in the animal kingdom in other ways as well?
Yes, as you go across the mammals, the thing that unites us all as mammals is breastfeeding and getting that milk meal. And certainly in rabbits, which is an animal this team in Dijon has worked on before, there is indeed a mammary pheromone, and that is crucial because a baby rabbit only has 3 minutes a day to get that day's milk, so it really has to find a nipple fast.
Wow, that's pretty impressive. I am going to open the floor for questions shortly, but I have a couple more questions in the meantime, but so get your questions ready if you're ready to ask any. So looking at, you've said pretty much that it seems to me all different sorts of species exhibit and utilize pheromones in a variety of different ways. And so one of the ways that we're talking here in terms of the title of our talk today is the scent of attraction, but what about as a repellent?
Well, the classic example, I guess, is skunks. So that can be a very powerful repellent, and that interacts with receptors in your nose in ways that you didn't want. So yes, that can work exceedingly effectively.
And is that a pheromone?
That wouldn't be a pheromone, only because pheromones are it's a signal within a species, but it's a very powerful signal. And one of the things that you'll know if you've seen a skunk starting to stamp is that it's a warning that it's going to let loose very soon. So that's the warning sign. Apart from its coloration, which really stands out, that's the warning to leave now.
Get going.
Yeah.
So I guess human beings have smell senses, and they're particular or dislike particular smells. In the animal kingdom, is it similar?
Yes. I mean, it's how animals find their food. It's probably how pheromones evolve. So our noses are very good, and this is true for animals across the animal kingdom. Our noses are very good at smelling the whole olfactory, the whole smell world. And if a smell becomes important for finding food, then we become more sensitive to it, and more of our brain is devoted to it. And that can be the beginning, if it's a signal, if it's a cue that's useful within a species, for male fish to start to follow female fish, and then females to start to produce more of those molecules to attract males, and then you've got a signal evolved. So yes, that's how it happens.
That's brilliant. I want to switch a little bit around and ask you a little bit about, I mean, one of the things that I mentioned in your biography earlier on was about that you founded the LGBT Staff Network at Oxford. Can you tell us about how that came about the environment at Oxford when this was happening?
So I arrived in Oxford in 1989, and there were two reasons for setting up the staff network. One was that there was lots going on for the students, but there was no informal network for staff if they needed support, perhaps if they were having trouble at work, and also to socialise. And the second reason was I'd arrived in the city and I was single. So if you want to meet people, start an organisation. And it started slowly. So I would go to the pub, having said that the group was meeting there. I was there with my flag, and I'd always take a book, because for the first couple of meetings I was the only person, but you'd have to stay until 9 o'clock just in case somebody came at the end. But now we are 200 or 300 strong. Wow. And it's fully recognised by the university. The Vice-Chancellor introduces the annual LGBT lecture during LGBT History Month, and it really has taken off. So I'm delighted by what it's grown into.
That's fantastic. There are student organisations, as you said and discussed, and in terms of Oxford, do you think that they've changed at all in terms of the environment?
I think it has changed enormously, in the sense that, well, walking here, the rainbow flag is hanging outside the Institute of Directors in Pall Mall. Not outside the Royal Society, but next year, I'm sure. But as you go around Oxford, the rainbow flag is everywhere. Now, it is only a symbol, but it actually takes a decision from those fairly conservative organisations to recognise that they have a diversity of staff and students. And I think it's very important as a symbol, and it does tell everybody that you're welcome. So that's the change that's happened even in the last 5 years.
Yeah.
And I think it's all going in the right direction.
Fantastic. You've attended, we have a series of events here at the Society called Out in STEM, and you've attended a few of those, I believe. And we talk often in those meetings about how being out as a scientist can affect their career. Has being gay, it sounded like you were pretty out pretty early in your career. Has it affected your career at all?
It's very hard to say. It's those things that happen in a decision meeting, whether you're getting a grant or getting tenure or an appointment or whatever, that you simply don't know. I have been fairly out, but sometimes at the beginning I was a little bit selective. And there was a moment when, out of the blue, my partner was coming from abroad, and suddenly I needed Sunday afternoon off, and so I had to come out to a colleague who I hadn't been out to before. Where I think it's had its impact is in decisions that I've made about careers. So Britain has been getting better and better through the years, particularly since '97. But there have been times when thinking about jobs abroad, for example, not applying for a job in Kenya, because I knew that if I was living in Kenya, I would be completely against the law. largely due to English laws that we left in the 19th century, coming back to bite us. But it would mean that I could never be relaxed. And similarly, whenever I go to a conference, there's that moment where you're wondering, should I take my partner or not? What's it going to be like at the dinner? Now, every time I've done that, it's been fine, and actually we've had great fun. So I think the message is actually just be bold.
Fantastic. Tell us a little bit about your relationship. I believe the person's here in the audience with us, so we won't point him out. How did you meet?
We were introduced at the Drill Hall at the theatre. It was a theatre group called Afro Pomo Homos, so African American postmodernist homosexuals. It was a great show, and that was 22 years ago.
That's fantastic.
Thank you.
I think that deserves a round of applause, yes.
And what's great is I still find it hard to say on stage my husband. And there have been odd moments, so when I have been on the radio, there's that moment where you say, a friend, or do you say your husband? So I'm getting better. And today has actually been one of those days, so thank you.
Fantastic. Are there any questions? If you do, we've got a roving mic over here on the side. Please raise your hand.
Hello? Yeah.
Yep.
Well, thank you for your talk. I just wanted to ask if you can give us like an idea of what it's like to work with pheromones from other animals, like what the process looks like. How do you actually get the pheromones? Do you filter the air? Do you actually purify them or something?
So how do you go about studying pheromones? That's a really good question, and that's been the thing that's been evolving. So, Budendant in Germany spent 20 years collecting from 500,000 silk moth females. And from that, he got 12 milligrams of material, which is just a couple of grains of sugar. Nowadays, with the change in technology, you could probably do that with a single moth. And the way that you do that is you can take an extract and put it into a solvent, into water or another liquid, and then you can run that through a machine called a gas chromatograph. And that basically separates the molecules so that, depending on which molecule they are, they reach the end of the long tube, 30 meters long, at different times. And then you can tell what the molecules are by sending those into another machine called a mass spectrometer, which will tell you what the molecules are. So that has revolutionized the whole way that you study pheromones, because now we can work on much smaller quantities of material, and instead of taking 20 years, you could do it in a couple of months, or even less. The other way that you can study pheromones is there are various molecular sponges that grab the molecules, and then you can release them when you heat the sponge. So they grab the molecules from the air and then release them. And there's been some lovely work on goats in Japan, where basically the male goats get very smelly, and they produce a lot of smell from their heads. So the and put the sponge under the shower cap, and then after a week or so, took the sponge away, ran the molecules through a GC, and they were able to work out which the molecules were that were both attracting the females and doing something I haven't really mentioned yet, is most of the time we work on behavior because that's easier to do the experiments with. So we know much more about pheromones that affect behavior. But pheromones also affect hormones and physiology, basically what the animals are doing in their bodies. So like producing eggs or whatever. And the male goats produce these pheromones which stimulate the female goats to start producing eggs again at the end of the summer. So that's the other way that you can study them. And the last thing you can do is that sometimes the amount of molecules, the quantity of molecules that are being produced is so small that we can't measure it using any of the machines that I've mentioned. So what you can do there is use the receiving animal's own detectors to work out when the molecule is present and even what it is. And that's been done most in moths. So male moths have these amazing feathery antennae, sort of like reindeer, but very fine. And these pick up molecules on their surface, pheromone molecules. And if you put an electrode at one end and at the other, you can record the stimulation of the antenna as the pheromone hits the antenna. And it will only respond when the right molecule is there. So you can use an electroantennogram to study molecules that are actually in such small quantities you can't even measure them on a machine. So there are all sorts of ways you can do that. Thank you.
Are there any other questions? Yep, at the back.
Really great talk. I was just wondering, have you ever done any switch studies where you take a pheromone from one species of moth, let's say, and then can another species of moth respond to that pheromone?
So the question is, can you do any, not almost bait and switch, but can you do any swapping of pheromones between species? So one of the things that has changed is with the silk moth, it was a single molecule that attracted the males. And what we discovered very early on was that closely related species of moth have almost the same pheromone molecules, but slightly different. So it's usually a combination of molecules that gives species specificity. It's why they don't get confused. And then there's something really neat going on. The males, these male moths, on their antennae, they have receptors for the molecules that their own female produces, but they also have receptors for the molecules that the wrong species produces, the other species. And if they detect the molecules coming from the other females, the females with the other species, they'll stop flying because they know that they're flying up to a dead end, as it were. Because if they arrive with the female, she'll reject him anyway. So they can tell whether they're flying up towards the right female. But the answer is each species has its own particular blend, and that's why they don't make mistakes. There's one twist to the story. There are only so many small molecules that animals can produce, given our biochemistry and what we can do. And somebody called Bets Rasmussen, who only ever worked on big animals, she worked on sharks and elephants. And she was working on elephants, and the female elephants produce a pheromone in their urine which drives the male elephants wild. But she couldn't find the molecule. And she teamed up with somebody who worked on moths, and it turned out this big animal, the female elephant, is using the same small molecule as about 140 species of moth. And so it turned out that the same molecule can be used by very different animals, but in that case, male moths are not attracted to elephants because the elephants don't produce all the other molecules that he needs. And male elephants are not attracted to female moths because the amount a female moth produces is so small he wouldn't even notice it. He's looking for gallons. So, yeah, thank you.
That would be quite an interesting match.
Yeah.
Just here on the front.
In your opinion, what is it about the elusive human pheromone that is making it so difficult to detect, whereas it's been done with comparative ease in other animals, for example, moths?
So what is it about human pheromones that makes it so difficult to find them? I think one of the hardest things is that you need a good behavior or physiological response to measure. And one of the things is that despite Masters and Johnson, we actually know very little about human sexual behavior, about what we do in private. And the other problem is that so much of our response is probably driven by culture. And so working out what is, as it were, innately human and what is learned behavior, particularly in relation to things like smells, makes it very hard. And there's one further thing, which is it's relatively hard to get money to work on sex because governments always get nervous. Mrs. Thatcher famously prevented that big survey when we needed to know about human sexual behavior when AIDS was at its height. And eventually it was the Wellcome Trust who stepped in with the funding to ask lots of people what they did in bed. So getting money to study sexual behavior is hard. The other thing is we don't seem to treat the sense of smell with the same seriousness as the other senses. So the example I always use is, in terms of civil society, We had a society to help people who were blind in the 1860s. The one to help people who were deaf was about 1911. The one to help people who have lost their sense of smell, anosmics, was only started in 2014. So it's only incredibly recently that we're starting to treat the loss of that sense with the seriousness that we treat the loss of the other senses. And I have a sneaking feeling about the way that most of us can still do our job, even without our sense of smell. If you're deaf or blind, it's much harder. So in principle, there's nothing that makes it really hard, but in practice, it's incredibly hard. But one day I think we may get there, but it'll take a while. One of the reasons I'm excited by the idea of the mammary pheromone potential, if it turns out to be substantiated in humans, is that that would give us the confidence to try something really difficult, which would be one of the sex pheromones. It's also possible that we don't have any. And then one last thing on that, which is that if you've ever seen the advert for Lynx, a deodorant favoured by young men, and the idea is that if you put it on, the lift will be full of, it was aimed mostly at heterosexual young men, your lift will be filled with many women, or if you're on the beach, you'd be sort of fending them off and having to swim off into the sunset. Well, the Lynx effect, I think, actually influences too many pheromone researchers, because what they have in mind is always a long distance effect. And it could be that if we did have a human pheromone that was involved in sex, that it was actually something that kicked in when we were actually getting much more intimate. So it might be more if other things were going on and we've gone quite deep into a loving relationship, since we're before the 9 o'clock lunch watershed. So it might be oral nasal, it could be all those kinds of things that it's actually acting on. And most of the experiments that have been done so far have been done at some distance and just by smelling.
So it may be that, in fact, the pheromones interact only when you're with the person who you want to be with already.
And getting up very close.
Yeah.
Yeah.
Very interesting. Are there any more questions? A couple. One at the back there and then two at the front here.
Sure, thank you. Advertisers spend a lot of money convincing us that smells are bad. Do perfumes mask pheromones, and is there any impact?
So advertisers spend a lot of money telling us that smells are bad and try to mask with with perfumes. Could those mask pheromones? One of the things about the way that smell works, and pheromones in particular, is that unless you can get a molecule that interacts with that particular receptor, it's very hard to block a pheromone. So if you've got a moth flying towards a female, you can fill the atmosphere with all sorts of smells, but it won't have any effect on his behavior because his receptors on his antennae are only going to respond to the particular molecules in the female pheromone. So it's as if the rest of the smell isn't there. So if we did have a human pheromone, the perfumes, unless they were targeted at that molecule receptor, probably wouldn't have an effect. My feeling is that perfumes are probably not related to pheromones. And the reason I say that is that perfumes change with fashion. So you can tell the perfume that your grandmother wore from the one that your daughter wears because the world has changed and different perfume houses are in the ascendancy. And that tells us that it's not a universal, it's something that changes with fashion. And what I would expect for something that has evolved over tens, hundreds of thousands of years, that it wouldn't be that fickle. So probably perfumes are doing something different, certainly something interesting, but it's probably more a learnt behavior.
At the front here.
I wondered if there have been pheromones discovered in great apes.
So are there any pheromones so far identified in the great apes? And the answer is no. And that would be a very good place to look. There's a group in Germany, that is looking at that at the moment, starting in zoos with captive primates, so with gorillas and with chimpanzees. And the advantage is that you can train the monkey to offer their hand and also to sniff. But so far, there's no evidence. And what we have is simply anecdotal. So when the male gorillas are thumping their chests, human observers have noticed a particular smell, and there are a couple of field accounts, but we have no idea what that means, and whether it's the smell or the sound that's really affecting other gorillas. So yes, that would be a very good place to look. Thank you.
Was there another question here? Yep.
Thanks so much for a really illuminating talk. You've spoken about the role that pheromones play in attraction between males and females of various species like dogs and elephants. My question was, has there been any research, or do you think there should be or could be, looking at pheromones as they relate to attraction or behavior between members of the same sex in various species?
So same pheromones acting in same sex. Well, there's a wonderful book which I think might be called Biological Exuberance, which is all about same-sex interactions across the animal kingdom. And there are the famous penguins with the baby penguin, well, actually an egg really, sorry, my biology is showing, being hatched by a pair of males. There are the wonderful stories of the gulls. So basically same-sex behaviour happens a lot more than was previously reported. And part of this is that biologists either weren't looking or it went in the desk drawer because they were either embarrassed or thought it hadn't happened. So the answer is that we know that it's happening all over the animal kingdom. And it's not impossible that same-sex interactions are being driven by the same pheromones. It really hasn't been looked at that much, and your question is a good one. We ought to be looking at that, particularly when such a behaviour is common. And we do know that with some of the long-lived birds, those same-sex interactions are very common. And if you're raising a chick, then you can be male or female, or two male birds, two female birds. So that could easily happen. We don't know if birds have pheromones, but I'm almost certain that they do, and that might well be a very good animal to start with. And then, yes, who knows? There's lots more to find out.
You said that you're not sure if birds have pheromones. Is that true generally? Are there any other sets of the animal kingdom that don't particularly you find have pheromones?
The other group of animals are all the mammals that have gone back into the sea. So they're air breathing, but they're back in water. So we know that fish do respond to pheromones. These mammals that have been on land and come secondarily back into water don't seem to have many receptors for smell. So they seem to have a very poor sense of smell. But for pheromones, all they need is the right receptors to have smell being really important. And there are some indications in a few of the toothed whales that who knows, they might have a pheromone, but that hasn't been investigated so far. One of the things that happen with birds is that when you look in the brain, the area, the volume of the brain devoted to smell is very small in most birds. But that doesn't mean that smell is unimportant. So one of the things that's really taken off in the last 5 years is looking at the sense of smell in birds. But as yet, apart from some work on some budgerigars and some amazing birds called crested auklets that smell of tangerines in the breeding season, and we don't know why, and actually there, both males and females smell of the same fruit. So we've got no idea what's going on. But we're at the beginning of understanding, and I think in the next 10 years, we'll suddenly discover, I think, that birds have pheromones along with pretty well every other kind of animal.
Fantastic. There was another question in the middle, and then over to on the other side at the back there.
So regarding, for example, perfumes, going back to that, and pheromones, if you have found your signature perfume and there's a certain biochemical reaction that happens when you found your signature fragrance, you may not actually be able to smell it, but those around you can. Has this to do with pheromones, or is this completely a red herring? And sort of along with that, So all those musk-based perfumes are just hooey?
Yes, two good questions. So one, I guess when you are getting when you're getting a perfume to match yourself, and that's one good argument for not buying perfume for other people, but giving a voucher instead. Let them choose if they're going to wear it. We don't really know what's going on with that. The effects are very subtle, and as you point out, it's all about individuals. It's all about signature. So that's probably not to do with pheromones, because that's individual smells, not the common smell of every male or every female. So far as the musk story goes, I don't really know. Again, whether musks are included in perfumes has changed. So some perfumes, older perfumes certainly do. One of the functions, I think, of the musks is as a fixative, a fixer. So it's one of the things that makes the more volatile components of the perfume stick around on your skin for longer. And it's something that male rabbits do in their chin glands. So there are some molecules which have a higher molecular weight, so they don't evaporate, and they stick on where he puts the mark, and so his signal lasts for longer. And the same kind of thing goes on in perfume. Now, whether that applies to some of the musks, I don't know. But the whole area of perfumes is indeed fascinating. And what they mean to the receiver, and also what they mean in terms of giving confidence when you splash it on before you go out in the evening. It's just a whole wonderful area of this whole interaction between smell, perfumes, and human behavior. Yep, thank you.
Just over there.
Thanks, fascinating talk. I just had a question in a similar line about perfumes in a way. When people wear a lot of perfume, they can kind of wear more and more to be able to smell it. And if you walk past a fish shop or something all the time, or you work in a fish shop, you stop smelling it. Given the sort of importance and specificity of pheromones, do animals habituate to them?
So do animals habituate? So this is when your brain actually no longer responds to them. As a rule, no. So something very special happens on the male moth antenna. As fast as the pheromone arrives, it's broken down. And he needs it, so there are special enzymes that actually break down the pheromone so that it stops giving a positive signal, because he needs to be ready to catch the next smell of the female. So habituation would be a real problem if he did. Generally speaking, that kind of thing happens in noses to, at least at the peripheral level, so right at your where the molecules are arriving, to make sure that everything is fresh, again, ready for the next signal. What can happen higher up in the brain is that if a signal stays the same, then eventually it does get ignored. But if it's something as important as a pheromone, and that if you ignored it, you wouldn't find a mate, then there'll be mechanisms to stop that happening. So, I think animals will always be on. The time that they're not, though, tells you a bit about their biology. So there are some male moths that at night are very responsive to the female pheromone. During the day, they don't fly because the female isn't signaling, but also they're at risk of being eaten by birds. There are some moths that once they've mated, the male moth has given so much of himself to the female, because as well as the sperm, he also gives us a protein donation, so a high energy and high protein donation. He needs a whole day to recover to produce more of it. And even if in the lab you blow female pheromone over his antennae, the antennae respond, but his brain says, no, wait till this evening. So you can have the control of response being either at the periphery or at the central brain level. And similar things happen in mammals too. Thank you.
Are there any other questions? Oh, we've got one over here, and then we'll come back in the middle again.
You talked earlier about anosmia and I did have experience of that. Does that mean that if we did, as a species, have pheromones, I would have been immune to it? Or is it something that's completely different from a sense of smell? And do other mammals species suffer from anosmia at all?
So a question about anosmia, and if you've suffered from anosmia, which can be temporary. So sometimes you have it after a very, very heavy flu or cold, and it basically knocks out the olfactory nerve cells in your nose, but they can sometimes grow back. Would that mean that you'd be unable to detect pheromones? Well, since pheromones work through your sense of smell, although there may be a special circuit, a special nerve, but it's actually in your sense of smell, the answer would probably be yes. Is anosmia common in other animals? And the answer is probably no, because if you were anosmic and couldn't smell the pheromones, then you simply wouldn't find a mate. So there's a very strong selection pressure against anosmia, and so you probably wouldn't leave any offspring. So the answer is probably no. But that's yes, the answer. Yeah.
I think we've got one last question here. We've got time for.
Thank you for taking my second question. You mentioned that in the moths there is a known receptor on the antenna for detecting pheromones. Is it known if there is such a receptor in the human nose, for example? And if we know that humans do have a specific set of receptor detector things for pheromones, could we not assume that humans therefore do produce pheromones? Otherwise, that would be redundant.
So a really good question asking about how we might study it from the other direction. Instead of looking at what humans produce, look at how they receive the signal in the nose. So we have about 400 receptors that still work. A mouse has 1,000 different receptors in the nose. We have about 400. But we only know what the molecules are that stimulate those receptors for a very few of them. Most of them are orphans, so we just don't know what the molecule is. Now, one of the twists on that androstenone story, which is one of the molecules I mentioned early on, which is not a human pheromone, but is often been claimed to be, is some great work done by Leslie Vossel in New York, looked at the receptor. And molecular biology had reached a point where it was possible to find this receptor and basically get it produced in tissue culture in large quantity. And you could blow the molecule over the tissue culture, and the tissue culture would basically shout. It would light up if it could detect the molecule. So, separately, she did some experiments where she asked people, what smell do you smell when you smell androstenone? And about a third of people think it's stinky, a third of people think it smells sweetly, and a third of people can't smell it. And she was able then to match what you could smell to the version of receptor for that particular receptor that you had. And it turns out, okay, so a lot of us can't smell it. Some of us think it is really stinky, and some of us think it smells rather sweet. And that's another reason why I don't think it's a pheromone, because if it was a pheromone, it wouldn't change. It would be one smell across all of us, and people who smelt it, as it were, wrongly, wouldn't leave any offspring. And so that's another reason why that probably isn't a pheromone, androstenone. But because we know so few of what the receptors are detecting, that's a long way off. One day we might, and what we'd be looking for is a receptor that didn't change, that was the same in all humans, that had been very conserved. And we won't know that for a long while. There are, I think, probably 300 probably more to identify before we'll know what the candidates would be. I think we'll probably find it from the other end first, and then finding the receptor will be the confirmation.
No more questions? Go on then. Very last.
What you've just said puts me in mind of coriander, leaf coriander. There are some people for whom it tastes like soap, and you mustn't put it in their food. And most of us, or I don't know even what the percentage is, think it's delicious and fragrant and marvelous. Is that could that possibly, or has it been discovered, whether that phenomena in any way applies to what you've just spoken of?
That's the perfect example, and we've only known it for 3 years. So basically, there's a group in New Zealand who have been looking at that very smell sensation. So coriander leaf, or cilantro for North Americans, is one of those love it or hate it smells or tastes. And the thing about what we conventionally call the sense of taste is really about smell. So it's a smell receptor in the nose. They haven't found it yet, but they've narrowed it down to a particular chromosome. And it's about 80% of us think that coriander smells wonderful and we can add it safely to food because it really makes it taste delicious. About 20% of the world's population, it doesn't matter where, so it's not about if you grew up in Indonesia, you're still about 20% of the population really thinks it tastes of soap. So enterprising restaurateurs in California now offer guacamole with and without cilantro because they know that 20% of their paying audience will hate it, so they can get the version without cilantro. And that's going to be one of the very first of many examples where each of us is in our own olfactory smell world. And it just amazes me that we can actually agree on anything like wine tasting, which is really wine smelling, because each of us, in the same way as we have different kinds of color blindness, each of us is in a completely different world. And it is an area that is just beginning to open up. And cilantro, coriander is one of the very first examples to be pinned down. The other is fuchsias. About 2/3 of people can smell that fuchsias have a scent. About 1/3, it smells of nothing. And that's a particular molecule, beta-ionone. Some of us have the receptor that detects it. Some of us have a version of that receptor that the molecule doesn't bind to, so we don't smell it. It's an amazing world that's just starting to emerge. Thank you.
One last thing to bring our two subject matters together, if I might, Tristan. You've talked briefly, I've seen you on occasion talk about a student who was transitioning, talk to you about hormones and the changing their smell. Can you tell us a little bit about that?
So it gives an indication of how the world is changing. I was talking to Oxford undergraduates last term, and I was talking about pheromones, and I talked about the human pheromone story. And at the end of the lecture, a student came up to me and said that they were transitioning, they were just starting hormone treatment, and they'd noticed that their smell was changing. And they said, well, wouldn't that be an amazing experiment to do? And it really struck me that 5 years ago, 10 years ago, I wouldn't have had an LGBT student coming up to me, engaging me, coming out, and actually saying, that's something that I've observed, and actually isn't that fascinating? And I just think it's an amazing measure of how the world is changing. So much further to go, pride awaits, there's much to fight for, but I think the world is getting a better place. So yes, perfect, thank you.
So I'd like to say thank you very much to Dr. Tristram Wyatt here. We've talked today about moths, goats, nipples, shower caps, sponges, smelly shirts, perfumes and peacocks. I think we should all go outside to Pride and see a few of those peacocks. Thank you very much.
Thank you.